In this video, we will discuss about which factor in the matrices contributes towards chemoresistance in the PDAC. As the heading suggests that it's the HA in the HELP matrices that modulates chemoresistance in the PDAC. So the researchers checked the effect of both HA and ELP proteins uh, on these pancreatic cancer cells, that which of them will cause this chemoresistance in the high HELP conditions. Since we know that HELP is the composition of two different kind of proteins. First one is the HA type of proteins, and second one is the elastic like proteins. So the data about this data about the study of these elastic like proteins is presented in the supplemental figures. Just for a brief to the audience, I will provide some of the insights. Since this ELP does not modulate the chemoresistance, they does not provide their data in the main figures. So what they did in the ELP is that in the ELP there is a domain, that is RGD domain, that helps in interactions with the integrin and CD44 that results in the mechanism signaling. So they hypothesize that it's that domain that might be playing a role towards mechanism signaling this pancreatic cancer cells. So they modified that domain from uh, RGD to RDG, and then use that modified matrices to check whether it will uh, prevent this chemoresistance in the PDAC. But they do not see any change in the modified high help matrices, that is with the RDG domains, compared to the normal RGD domains. So from that experiment they come to a point that ELP does not modulate chemoresistance in the PDAC. Then they check for the HA modifications. So in this case of HA modifications, what they did is they replaced the HA with this inert 8-arm PEG, which is known as benzaldehyde modified PEG, and they check its stiffness uh, with the help of shear modulus measurement, and they find out that it low help has a lower stiffness and high help has a higher stiffness. This is what they have expected. And when they check this chemoresistance in this PDAC, and they can see that in the, they can not chemoresistance, they want to see the organoid growth in this microscope. They can see that organoids are growing both in the ELP PEG high conditions and the ELP PEG low conditions. And when they check the expressions for this ABCG2, they can see that ABCG2 are expressed in both the cases, and the difference is non-significant, indicating that this this alterations of this HA results in the reduction in this ABCG2 genes in the tumors, and this HA might be the reasons that triggers chemoresistance in the PDAC. Moving forward, they checked this impact of these gemcitabine drugs on this modified PEG, the benzaldehyde modified PEG, and this ELP high conditions. And they can see that both this ELP PEG low and ELP PEG high, even after passage force, does not show a significant change, both in the case of cell viability and also in the case of cell toxicity. The changes are not visible. And when they quantified these results, we can see in the HA tag, when the HA or help matrices is used, they can see that increase in the IC50 requirement to reduce the biological functions by 50 degree, or we can say the IC50 gemcitabine requirements are higher when they use this help or these high help matrices with HA. And when they use this ELP PEG high, they can see that reductions in this gemcitabine requirement to reduce the biological functions by 50%. So this further substantiate their previous finding that it's the HA tag it's the HA in the help matrices that contributes towards this chemoresistance in the PDAC. And when they check this organoid, when they this panel F is the results on the single cells, and when they did the same experiment on the organoid with passage 4, they see the similar results, that HA contained help high matrices requires more gemcitabine for IC50, whereas PEG ELP high requires low gemcitabine I50, indicating that it does not have the chemoresistance. So all these results indicates that it's the HA in the help that helps in the chemoresistance in the PDAC. So targeting HA in the tumor microenvironment would be the new effective therapeutic target. But this is not what they have concluded. This is just from my point of view. We will discuss their further experiments in subsequent videos. So if you need to understand more about their experiments in detail, you can go through the reference that has been provided in the description. So that's all for this video. Thank you for your time.